My name is Teresa Gunn. I am the president of the International Mammalian Genome Society, and it is a really great pleasure for me this evening to introduce the Verne Chapman Lecture this year. For those of you who don't know who Verne Chapman was, he was one of the founding members of our society. He was very collaborative, and as I never got to meet him, but from what I've heard, he was interested in lots of different science and saw mouse genetics as the tool to learn a lot about biology. And I believe that he and our speaker, Joe Nadeau, were good friends. And Joe, am I right that you were also a founding member of the society? Yeah, so, and our speaker, Joe Nadeau, he is, again, one of the founding members of the society. I forgot to look up his CV, so I told him earlier I can't tell you all about his magnificent distinguishedness, but I actually first met Joe more than 20 years ago when I was a graduate student, and I strongly considered doing a postdoc in his lab, but I had left Montreal 4 years before and wasn't ready to go back, and that's where he was at the time. So Joe is now at the PNRI, Pacific Northwest Research Institute in Seattle. He's been there for several years now, and he is full of magnificent distinguishedness. And so I will just let him tell you all about his research. That's the best Verne Chapman introduction ever, isn't it?
Absolutely. I don't know where to go from there. Whoops. So this is Verne, in case you don't know him. A long time ago, when I was in 7th grade, I used my allowance to buy a genetics textbook, and I was hooked. I loved genetics. And then when I was an undergraduate, I got to participate in the undergraduate research training program at Jackson Laboratory, and I worked with Roy Stevens. And I loved mouse genetics. And then after a postdoc with Jan Klein in Germany, I came back, and I thought about doing a postdoc with Verne, but he was in Buffalo. And you know, you like Buffalo, but Buffalo is Buffalo. So I ended up at Jackson Lab, but we became not only good scientific colleagues, but also really, really good friends. He was smart. He liked to get things done, not just talk about things, but to make things happen, and always in a collegial, inclusive way. And a lot of the reason the society is here, the journal is here, is because of Verne. With Jan Klein, Lee Silver, Peter D'Estachio, Steve Brown, and some others, we started the society, we started the journal, and we wanted to build a community where people worked together so that we had the infrastructure to do more than we could individually. So by working with Verne and Jan and Lee and others, we discovered this whole great community of mouse geneticists. So it began with genetics, mouse genetics, and then the people who do mouse genetics. And it's been incredible. So as a little bit of preamble to what I want to talk about, sometimes you do experiments and you get sort of what you were looking for. And when that happens to me, I get nervous. Because I always think that means I wasn't thinking hard enough, smart enough, clever enough, or nature's tricking me. That it makes it look simple, I'm simple, and there's something more subtle going on. Sometimes we're wrong, and that's really exciting because then you know you're making progress when you don't go anywhere. Sounds like a Yogi Berra thing. But the best times, the ones I like most, are when we stumble on something that's completely unexpected. I'm going to tell you about some of those tonight, 2 of those stories. And someone asked once, why did you look for that? And I said, are you kidding? You never look for these kinds of things. They come along. You just have to be lucky enough, whatever, stubborn enough, something, to recognize something cool is sitting in front of you. And sometimes it takes a while. It's like a Homer Simpson moment. There's a duh. In one case, we had to it's good to include controls in your study. It's even better to pay attention to the controls. Okay, so in 2 parts of the lab, and Jason Heaney, who's here, was part of my lab at the time, we were studying two different problems, testicular cancer and neural tube development. And part of the story is what you missed if you would have come to Montreal. But Montreal was complicated back then. And both projects stumbled on the same thing that had a regular interpretation and an irregular interpretation. And the regular interpretation we read about, see in the papers, the journals all the time. The irregular one was really irregular. And everybody looked at the data, and it was a really good group at the time. People were critical in a really constructive way, but they wanted to see the evidence, they wanted to think through the logic, they wanted to be convinced. Both sides were convinced that the irregular interpretation was probably right, but nobody wanted to write it in their papers because you're graduate students, you're postdocs, you need the papers, you need to be provocative, but without going over the edge. So a few years ago, Gunwa Nkuzi, the graduate student, said, well, let's write this up, but let's not write it up as the conclusion, let's say it as a question. And so is it the regular or is it the irregular? And we're convinced it's the irregular, but this is where you guys come in. I've presented this a couple of times. And if I've missed something, if we've missed something, somebody in the back row stand up and say, this is wrong, you forgot about, and the simple explanation is, and if that's right, and we've been thinking about this in the wrong way, then that's great. We'll put a stake through the heart of this. We'll never talk about it again in public. But so far that hasn't happened. So the obligation is on you to either make or break this. Okay, what did we do? Nothing. There we go. We skipped one. There we go. So, sperm and egg come together, fertilize, make a conceptus. The conceptus makes an embryo. The embryo grows up to be an adult. We go through this cycle of mating and next generation. And we know lots about the biology of this. But part of the fun in research is that there's a whole lot more that we don't know than we do know. We take pride in what we know, but I think there's a whole lot more that we haven't discovered yet. Then this cycle and the molecules that are inherited, that transmitted from one generation to the next, not only propagate the phenotypes from one generation, but also the variation from individual to individual, phenotypic variation as well. And Mendel and a lot of others at the time were fascinated by this. And Mendel, as you know, did his crosses and worked out the rules, his laws, the rules of segregation, assortment, and dominance. And he worked them out in a statistical sense. We now know the molecular basis, the DNA sequence and the variation, and we live and work with all that. In fact, we know those rules of inheritance so well that we sometimes take them for granted. And there can be crosses, data sitting in front of us, and we don't think about whether Mendel's law correctly applies or whether things are following these laws in the way they should. We just take them for granted and keep on going. And I'm going to give you some examples of this. The other issue here is that his rules of segregation in a heterozygote producing two kinds of gametes, until recently we couldn't really look at the gametes directly. We would see the gametes as manifested after fertilization through development as the way they show up in the next generation, and you work back from the ratios of 1 to 2 to 1 from an intercross, the 1 to 1 in the back cross, to what the gametes must have been. But that little window in there creates some opportunities for troubles. Whoops, this thing's going to drive me nuts. Okay, so AB heterozygote transmits alleles in equal frequencies, Mendel's law, 1A to 1B. In a back cross, 1AB to 1BB. You guys all know this. An intercross of 2AB individuals, 1 to 2 to 1. This assumes that the gametes come together at random. If they don't come together at random, then you don't necessarily get these ratios. We know some exceptions, SD, the chromosome 2 locus that Fernando and others reported recently, T haplotypes. There's a few exceptions, but in a way the exceptions show that most times, in most places, Mendel works. But what do you conclude in a case like this? Say we made a knockout. The way Alan Bradley pioneered some of this. Make a knockout, do the intercrosses to see what the homozygous mutant looks like. What's the phenotype? We expect a 1 to 2 to 1 ratio, but say you see 1 to 1 to 0. What do you conclude? Well, the homozygote is embryonic lethal, half the heterozygotes, a dosage effect, so we write the paper. And there's lots of papers like this. So the deficiency, all homozygotes missing, half the heterozygotes. Is it right to say it's lethality? What you'd expect, if that's true, is that the litter size should also be reduced by half. If you're missing all the homozygotes and half the heterozygotes, then you're missing half the genotypes, and if they died, the litter size should be reduced. What if the litter size isn't reduced? Then what? Because there's I'm going to tell you some examples of this. And so this is what we wonder about. Do gametes woo? In other words, we think of them coming together at random and mating, and you see these pictures of egg and a dozen sperm. That turns out that only happens in vitro. When you have an egg in addition, you dump sperm on them. In vivo, it's a long, convoluted, tortuous path to get to the egg, and not many sperm do. But is there a preference then in the combinations that like each other? And that's where we're going with this. What's the evidence for that? And again, if I'm wrong, tell me, we'll put an end to this. So we were working on testicular cancer of the germline. The germline is really interesting in that for most of its development, it's unipotent. It's going to make sperm in a male and eggs in a female, and that's all they're going to do. And yet at fertilization, it does this remarkable switch from unipotency to pluripotency, because now you have to make an organism. Then the cells are set aside early in development for the germline of the next generation. Maintaining, controlling pluripotency is really important, because if you don't do that, you've got problems. You don't make the right kinds of gametes in the right numbers, and you can get a testicular germ cell tumor. It's the most common cancer in young men. It's the third most heritable cancer, has an unusually high rate of metastasis. It's readily treated, like better than 95% long-term recovery. But among those individuals that have recovered, and we say readily treated, chemotherapy, radiation, near-death experiences to live, which is a good thing, but we haven't really treated it if that's what you have to go through to recover. They're also at higher risk for secondary diseases, metabolic disease and atherosclerosis. These tumors make muscle, skin, hair, bone, cartilage, and they still retain some embryonic stem cells usually. These were discovered by Roy Stevens at Jackson Laboratory, as I said a while ago. And he discovered the mouse model, and they have a developmental origin. And we know something about these tumors that we don't about many other tumors. We know the cell, the location, and the time. The stem cells are primordial germ cells. The critical period is around embryonic day 12, give or take a day or so. And we know where they are. They're in the urogenital ridge, the part of the embryo that will eventually make the testis. That critical window, there's 25 or 30 cells, primordial germ cells. A couple of them will transform into tumors, and that's it. If they get through that window, they're fine. The genetics, it's in the 129 strain, but not other strains. When we see that, we say, it's genetic, because it's restricted to, occurs only in one strain, but not other strains. So Roy did what any good mouse geneticist would do back in the day. We would do it a little bit differently now, but it's still knowing what he knows, what he discovered, we probably wouldn't. He crossed 129 with other strains, made F1 hybrids, intercrossed the hybrids, and looked at F2s, and looked for inheritance of cancer susceptibility. Do you know what he found? One animal affected among 11,000 male offspring. So it's clear it's genetic, and it's clear it's really complicated. Either there's a lot of genes or some other kinds of funkiness that complicate. But it's nothing any graduate student, postdoc, or granting agency would ever do to pursue something that segregates at the rate of 1 in 11,000. And it's also the first evidence for the stem cell origins of cancer. He and Barry Pierce demonstrated this. There had been a theory about stem cell origins. Roy and Barry provided the first experimental evidence, the first that I know. So we thought, if we can't get at the genetics of 129 directly, maybe by working with modifier genes, we can identify those, figure out how they work, and work our way into the genes that act in 129 to control susceptibility. The first one we worked on is the gene that Roy and Noguchi described called dead end. The mutation was called TERR for teratoma. In a wild-type homozygote, let's see if I get the mouse. Nope. I give up. Wild-type homozygote, 7% are affected, just like in 129. Heterozygote, 35-ish%, and a TERR homozygote, 95%. So this is a modifier. It's not required for susceptibility, because 129 gets the tumors without, and it's not sufficient because on other backgrounds it causes sterility, but no tumors. So we cloned it. It turns out it's a point mutation, a stop mutation in the dead end gene, previously described in zebrafish as a germ cell factor, but not a tumor gene. And the mutations is in a really interesting motif called harama. It's not, this motif is not in many genes, but the genes that have it, it's really, really well conserved back to bacteria and other kinds of wee beasties like that. So this is the dead end tergene. So we made a knockout, and we intercrossed the knockout. And what you see here in the second and third line, the back cross maternal means mother's heterozygous, back cross paternal, dad is heterozygous. Let me see if this works. Yep, 1-to-1. Wild type heterozygous. I'm not listing the homozygotes because they don't occur. Okay, so 1-to-1, 1-to-1, Mendel's happy. Look at the intercross, 120 to 162. Now, if the wild type number is right, we would have expected 240 heterozygotes. So there's a significant deficiency. First issue. So we say, okay, well, they're embryonic lethal because we don't see them. We flushed embryos from the ovum. We work backwards through development. We get to embryonic day 3.5, there are no dead embryos. Second point. The third point is look at the litter size. It should be about 2/3 here of what it is there, except it's indistinguishable. So how do you lose 1/3 of the embryos? You don't find them dead, and but not affect the litter size. So the back crosses are important. They show that gametogenesis is normal, producing the right numbers of gametes in the right numbers. So back cross to wild type, from either heterozygous male or female, everything's fine. It's only in the intercross. So in a way, the back crosses are controlled. Okay. This shows dead end, the results here. This is Argonaut-2, something we just finished studying, and the paper's under review. It's involved in the RISC complex as part of the catalytic subunit in microRNA biology. The two back crosses, maternal heterozygosity, paternal heterozygosity, 1-to-1, pretty much 1-to-1, everything's good. Here's the chi-square tests are not significant. We tested both 1 to 2-to-1, and then with the other degree of freedom, we tested 1-to-1, because sometimes this is just the absence of homozygotes distorts the results a bit. Here's the litter sizes. In the intercross, we see 1-to-1 to 0. Now we know, it's known from other labs, that the homozygotes are missing. So it should be reduced by 1/4 for this. So that's maybe this difference, but it doesn't account for what's the missing heterozygotes. So in some cases, in both of these, dead end and Argonaut-2, there are too few heterozygotes without affecting the litter size. But there's another class, too many heterozygotes. This one was in the literature from our collaborators, Nick Davidson and Valerie Blank at WashU. They made a mutation in the ACF genes, the ApoBec complementation factor. We'll get into the biology a bit. In their case, they only did an intercross, 31, 301, 0. So they got 10 times more heterozygotes than you'd expect. If you look and this is a mixed background. They gave us the stem cells. We remade the mice, kept it on a pure 129 background. Maternal heterozygosity, paternal heterozygosity, 1-to-1, 1-to-1. Litter sizes, intercross, 75, 229, 0. Here's the chi-square test. But look at the litter size. It didn't make a difference. Now we're going to come back to some of the interpretations and the arguments to help sort out what might be going on. Here's DDX1. It's a dead box helicase, encodes an RNA helicase, intercrosses. We're going to get into more of the details with this one in a minute. But again, 42 to 366, 34 to 258. Way too many heterozygotes. In this case, they argue that heterozygosity for this engineered mutation is killing wild-type animals. So it could be, but you'd like to see some additional evidence for that. Palmilio-1, this is this paper here that describes it as a loss of pre-implantation embryos resulting from this gene trap. Back cross, intercross, chi-square tests, litter sizes. So I wrote to him, did you have the litter sizes weren't in the paper. I said, was the litter size changed? Did you measure it? Yeah, it didn't change. I said, did you look for embryos early in development? And we didn't see them. So how do you reconcile that? So what we think is happening is in a heterozygote for some of these, and I've drawn this heterozygous as the male, it could be the female, they've got one choice, an egg. So they're going to fertilize in equal ratios. It's as though there's a predisposition that the sperm are carrying, but when given one choice, they do what they got to do. In an intercross of these two, and normally, you get all four combinations in the 1 to 2 to 1 ratio. If it's lethal, you miss this group, but you keep these, but the number of embryos should be reduced proportionally to the lethality. But if it's biased, these sperm and these eggs have preferences. So all the eggs are fertilized, but they're fertilized in a non-random way. So that this sperm, given a choice, will like that egg. This sperm, given a choice, will like that egg, but not that combination in this cartoon. This is the DDX story. It's a little bit more complicated than that I just described. So they dug out their litter size data for us too. And in this case, they made a knockout of dead DDX, and it reduced litter size, and they lost some wild-type animals. But the loss of wild-type happened in some crosses, but not other crosses, so they call those the star animals, because that parent that carried that knockout, like it was a paramutation, would lead to fewer wild-type offspring. So some families did, some families didn't. And you can see all the different crosses. These are the control crosses, and then these are all the different test crosses, and you can predict from their model what the loss should be. So when they shared the litter size data, it hardly changed. Now this one's a bit lower, but here when you're crossing these that shouldn't produce anybody, practically, 5.5 isn't so bad. Now the last example is one of the more complicated, but to me one of the more interesting ones. Vicki Nelson was interested in the effects of ApoBec1, and we'll see why in a minute. And she got a knockout from Nick Davison and Valerie, and she crossed it with dead end TER, wanting to know whether these interact to control susceptibility in some way. We'll get back to that story in a minute. But here, when she did, mom is heterozygous for the dead end TER mutation, and dad is heterozygous for the ApoBec knockout, you get 27, 30, 25, 32 kids, the double heterozygote, each of the two single heterozygotes, and the wild-type animals. Mendel would be happy, 1 to 1 to 1 to 1. Look at the reverse cross. When mom is heterozygous for ApoBec and dad is heterozygous for the dead end mutation, 64, 19, 19, and 13. So a huge deficiency. All of this is done on the defined background. All the studies I'm telling you about are done on an inbred background. The only genetic variant that's segregating is the one we're talking about. Litter sizes here and here aren't different. So this looks like lethality. So somehow, remember, we have a dead-end TER mutated colony by itself. We have an ApoBec knockout colony by themselves. So we know what those do on their own. But here in this cross, they do something different than they do separately. There's some numbers that go with this. If this is the right number for wild type, for the sake of argument, then we would have expected 64, 64, 64, 64. We only see, or a total of 192, we only see 51. So we're missing 141 animals. We're only seeing 27% of what we expect here. So there's a lot of embryonic loss and no change in litter size. And what's interesting is that not only are we missing these animals in this cross, they're heterozygotes, which are the same genotype as the parents and is the separate colony that we use for breeding. And dead-end TER homozygotes are fully viable. Not the knockouts, but the TER homozygotes are fully viable. ApoBec knockouts are fully viable and fertile. So how do you create a circumstance where if you want to argue lethality, where these embryos are missing and because they're lethal, when the parent homozygotes are fine, and in separate crosses they're fine? The argument is that because the litter size didn't change and because of these other arguments, the sperm and eggs are making a difference, and ApoBec in mom and dead-end TER in dad are influencing determining factors in gametes that lead to this gamete preference. These are the genes that woo, that we've reported, ACF, ADAR haven't talked about, and protein phosphatase 2C beta. This evidence is a bit circumstantial, but the others are pretty firm. What's interesting is all the ones in red, we've also shown affect testicular cancer risk in a heritable epigenetic manner, in a transgenerational manner. So these genes not only dictate gamete choice, apparently, but they also affect cancer susceptibility in offspring that don't inherit these cancer susceptibility genes. In other words, the kids are wild type. By the genome sequencing, you should be able to predict the risk from what we know about their DNA. Yet the risk isn't determined so much by their own DNA, but by genetic variants that acted in ancestral generation that presumably induce an epigenetic change, and that change is inherited and affects cancer risk. So we know how to induce this, we know how to propagate it, and as I'll show you, we also know how to reverse it. if I can go back one. But there's a serious ascertainment bias in this list. A lot of these genes are RNA genes and fall to 3' UTR, RNA editing, microRNA biology, RNA stability, translation control. But those are the genes we've been thinking about. So we really often go back through the literature more systematically to see whether it really is about RNA biology or whether there's other parts of the gamete physiology that we need to consider. Okay, the number of eggs that's ovulated, where does the litter size come from? The number of eggs a female ovulates largely determines the litter size. And she ovulates before fertilization, spontaneous ovulation. So when she's ovulating and producing eggs, she doesn't know the genotype of the sperm that will be on the way later. She completes the second meiotic division, the equational division, after fertilization. We're going to get back to this because there might be an interesting twist. And the reductional division, the one that's determining which of the maternal alleles is going to be in the egg and in the oocyte functional, is determined at the first division, which was back when. So there's a trick to this. How does this work? Because all we've said is that it happens near fertilization and when the egg and sperm are there, but the critical events determining the genetics were sometime before. So here's meiosis in males. I've got a 2N genome, and you go through the reductional division, then the equational division, you get the 4 sperm, you know the story. And in females, same case, you get the reductional division first. As you can see here, this is the polar body, that will be the egg. Sperm comes in, you complete the second equational division, and you get the egg in the second polar body. But notice when the sperm comes in this model, the egg and the polar body are the same. The sperm isn't going to dictate one way or the other, one outcome versus the other. But there was a recent paper in Nature Genetics by Ottolini looking at this issue in humans, looking at the egg and genotyping the egg and the polar bodies. And what they found is that in many cases, meiosis is reversed. The equational division is first, and the reductional division is second, after the sperm comes in. And in this case, then, the sperm could come in, bring some information that's critical, and drive one of these to the polar body and keep the other in the egg. And so if this happens in mice, if it happens often, if these genes are involved, this could be a way of driving gamete preference. This is part of the fun of biology. We're taught this, and that with the tools we have now, we can test whether the rule is really true or whether something else is going on. Okay, neural tube defects. This is the other part of the story. One of the most common birth defects, 1 in 1,000 births. It can be suppressed with dietary folate supplementation if you're a young woman or if you're a husband and you're planning kids. Everybody knows some extra folate in the diet is recommended because you can suppress neural tube defects, about 50%. So 50% of a big number, of the most common birth defect, is a big number. But 50% are resistant to the beneficial effects of folate. So 50% of that big number is still a big number. So we were interested in what's different about cases that are resistant to the beneficial effects of folate versus susceptible. So we took some models and we tested folate susceptibility, and we focused on several. But what's interesting, there's more than 200 single gene neural tube mouse models. Very few are tested for the response to nutrients, folate, methionine, thymine, and inositol. That's about all I think that's been tested. And you can see these are the genes that have been tested. Most have been tested for folate, but not all, and then the others is sporadic. It would be really interesting and really important to systematically go through these different nutrients and the different models and figure out who's susceptible, who's resistant, and the mechanisms of benefit versus no change. So Gunwa used 2 mutants, apolipoprotein B, knockout, and the loop tail mutant. And the protocol was this. Before pregnancy, she put mom on a folate supplemented diet with either 2 parts per million or 10 parts per million folate. 5-fold difference, well within the normal physiologic range. And then, as embryos, she dissected them between embryonic day 12.5 and 14.5, phenotyped them and looked for incidents of neural tube defect and correction, and counted embryos and live or dead. Okay. In some cases, the supplementation is done during pregnancy, which is fine, except it doesn't really get at that issue, because this issue of gamete choice, because That's already happened. So it's only in the preconception introduction of the dietary supplement that makes a difference. So these are the results. Here's apolipoprotein B. Again, all in the same background, the same segregants, the same crosses. The only difference is 2 parts per million, 10 parts per million folate. So in apolipoprotein B, wild type, heterozygote, homozygous mutant, 24, 48, 27. Perfectly good Mendel. Look here, 40, 38, 1-to-1, and fewer homozygous mutants. There's the chi-square value. This is the percent of loss. We're missing about 53% of the heterozygotes and about 30%. So you're missing a lot. Look at the litter size. Didn't change very much. This loop tail effect is strong if you measure it in terms of effect size. The chi-square test is sort of in the suspicious range, but not statistically significant. But again, a lot of embryos are missing, no change in litter size. So there's a genetic effect through the genes I talked about earlier, and there seems to be a gene-diet interaction. These genes exposed to different amounts of folate in the diet. Okay, there's some other genes that do this. We went back through the literature. Now in this case, these two studies we talked about, and Lee Niswander talked about the detrimental effects of folate, that putting folate in the diet is actually killing embryos. Now this is back in the early days of Obamacare, when people were against the government getting involved in healthcare because there were going to be death panels. So we were nervous about writing this interpretation, because folate is, by the CDC, is supposed to be in our diet. So if it's killing embryos, that means the government's putting something in the diet that's killing our babies. So we didn't want to go there either. But it turns out it's probably gamete preference. So it's interesting, because if that's the case, in humans, if something like this happens in humans, we see fewer cases of neural tube defects. We assume that means because the folate, the additional folate supplementation, fixed a developmental problem. Maybe it didn't. Maybe in some cases it does. Maybe in other cases it influences the number of cases that might occur in the first place. And if they don't occur in the first place, you never see them later, which from a public health viewpoint is fine, because you don't want babies to be born with this often life-threatening, debilitating condition. But you'd like to understand why it works, how it works. So the effects have to be haploid. If they were at the diploid stage, you wouldn't see this difference between gametes. It could be sperm competition in egg drive, that you're producing one egg versus another. One sperm has an advantage over the other. Those are ideas that are testable. It could be gamete attraction. Imagine back in the day, you've got an egg, there's sperm looking for you, and not many are going to find you. You're hard to find, and it's brownie in motion, they swim. But imagine an egg figures out, I can send a signal, a little beacon. I'm over here. And sperm have a receptor for that, can recognize that. Say, I'm coming, I see it. So sperm-egg interactions, receptors, ligands, olfactory clues, some kind of chemical sensing would have a tremendous advantage over combinations that don't do that. And then just after Gunwa and I submitted the revision of the paper came out in Nature about a receptor on the egg for a ligand in the sperm, and the receptor is called Juno, and it turns out it's a member of the folate binding receptor family. It doesn't bind folate, but it's a member of the family. So is that a clue to what's going on? It has to happen before the acrosome reaction is triggered, otherwise the egg is determined. Okay, what do we think is going on? I suspect that polyamines, for sake of argument, are part of the biology here. There's at least 4 kinds of polyamines. These are nice, simple chemical molecules: spermine, spermidine, cadaverine, and putrescine. I don't know why. There's putrescine. So these are the molecules that make cadavers smell putrid or like a cadaver. And they're also the molecules that make semen smell like semen. There's a lot of this in the cell. There's 90%, the cells are loaded with this. 90% of them are attached to nucleic acids. They're involved in a lot of different functions. What's the connection? Here's the folate metabolism. 5,10-methylene tetrahydrofolate goes through this cycle, transfers the methyl group onto homocysteine to make methionine. That's adenylylated. That's the methyl donor for all of the methylation reactions of DNA, lipids, proteins, whatever. Methyl group is transferred to other substrates. You make S-adenylyl homocysteine, you do this cycle. Choline and betaine are alternative methyl donors. So we know this part really well. Connected to this, though, is the polyamine pathway. Ornithine decarboxylase to putrescine, spermine, spermidine, and you can make them and you can catabolize them. S-adenosylmethionine is an alternative substrate for this pathway. What's interesting is that when a cell is stressed, there's at least 2 papers on this, when a cell is stressed, it doesn't have enough folate. What does it preserve? Polyamines. It gives up methylation or reduces methylation to keep polyamines. Now we all, as geneticists, we all love methylation. We all say it's really, really, really important for all the reasons that we know about and study sometimes. The cell is saying, it's important, but if I'm preserving polyamines, I'm telling you the polyamines, whatever they're doing, are more important. So under these conditions, maybe the cell is using this. Now, acetyl-CoA is also the source of choline and betaine. So as this is catabolized, it's eating up a lot of acetyl-CoA. And so it's compromising folate metabolism in 2 ways, by using SAM, the methyl donor, and by using up the alternative folate methyl donor substrate. These are some of the pathways, the functions that polyamines are involved in: necrosis, apoptosis, cell proliferation, gene regulation. They bind to DNA, they involved in DNA acetylation, cell programming, autophagy, translation, lots and lots of functions. The connection between that and the biology isn't so clear, but there's enough going on to be suspicious. There's also this: if you're deficient in polyamines, you tend to be infertile. If you supplement with SAM, S-adenosylmethionine, or polyamines, you restore fertility. If you overexpress ornithine decarboxylase, and that's the other source of the polyamine synthesis, that leads to infertility. It's argued that ornithine decarboxylase is perhaps the most regulated enzyme in the body, because too little polyamines is really detrimental. And there are inhibitors of the inhibitors of inhibitors for polyamine, for ornithine decarboxylase. In gametes, they're very highly regulated. This is OAZ3 is a testis-specific inhibitor of ODC1. OAZ3 deficiency leads to aberrant sperm that don't fertilize because the tails fall off. Azin, which is an inhibitor of OAZ3, is also expressed in haploid cells. So what are these genes doing in haploid cells that's guiding the biology? And could this be part of what's going on? When we did gene expression profiles in Gunwa's study, and then in this other study from a group at McGill, these polyamine genes were differentially expressed with and without, either in the neural tube models or with folate supplementation. So something's going on, and there's ideas about how to test this. Okay, so questions. Genetics. Is this another manifestation of epigenetics? Is it gamete preference? Folate effects. Is it limited to folate, or is this a general response to nutrient deprivation, say through mTOR? Fertilization. Is this an attraction? Is this an interaction before triggering the acrosome reaction? And then obviously, if you are biasing the genotype distribution from one generation to the next, there are obvious population, genetic, and evolutionary implications. Okay, now we're going to switch gear to the bigger story about epigenetic inheritance, and then we'll finish with that. So to me, epigenetic inheritance is the inheritance of phenotypes, just like Mendel was interested in phenotypes, through the germline and independent of DNA sequence differences. So we say through the germline because you can inherit your political affiliations from your parents, inherit in the soft sense. You can inherit your religious inclinations through your parents. Apparently even your happiness is inherited in that kind of way. But through the germline and independent of DNA sequence differences. So this is about phenotypic inheritance, the origins of phenotypic variation, independent of DNA sequence differences. The triggers can be environmental agents of various kinds, and they can be physiologic stresses to genetic mutations that we've inherited. A lot of us, if you do the catalog of mutations that we have, like Yuichi talked about today, and that's been in the literature, and Jim Crow talked about too, all of us have more detrimental mutations, probably 100, 200, than we would expect given our health. We're healthier than our genetic content would imply. And you could imagine that in response to that, your genome does epigenetic changes to accommodate that and to try to get closer to optimal function. And so these genes can be involved in either creating a stress or in the mechanisms that lead to epigenetic molecular inheritance. So testicular cancer is also a good model for this. In humans, there's a 4 to 6-fold increased risk of father to son. So if your father's affected, you have a 4 to 6-fold higher risk of having testicular cancer than if your brother is affected. And if you think about the genetics of that, it doesn't quite make sense. Sprouty 4 increases risk but only to testicular cancer, but only through the maternal germline, not through the paternal. There's sex-specific methylation. In mice, we're going to talk about this a little bit, transgenerational epigenetic inheritance, variants that have conventional effects as well as multi-generational effects. And then parent of origin effects here with ACF and Argonaut2. Okay, when we cloned dead end, not long after that, Reuven Agami at the Netherlands Cancer Institute in Amsterdam noticed that in some cells the microRNA level and the protein level is high. We expect this reciprocal relationship. One is high, the other is low. But in some cell lines, in testicular cancer in particular, microRNAs are high and the protein is high. And he speculated, he hypothesized that maybe there are RNA binding proteins that bind to the target site in the 3' UTR of transcripts and block access of that microRNA to its target, mRNA, and therefore allows that protein to be produced even though the microRNA is present in the cell. It's a level of microRNA control. So he tested a panel of RNA binding proteins, and guess which one was responsible? Dead end. So it blocks these microRNAs from these mRNAs and allows them to be still produced. Notice it also protects itself, which is cute. So this is the model. At the same time, when we did the sequence analysis for dead end, it turns out by sequence it's related to ApoBec complementation factor. And what's that? That's the RNA binding subunit for ApoBec1, the cytidine deaminase that converts the C to U that's read as a T. So ACF brings ApoBec1 to target sites in mRNAs, and then the ApoBec1, the cytidine deaminase, does its editing. And dead end is related to this. So we had some hypotheses about dead end, about the microRNAs, and about ApoBec1. And those are the 3 last parts I'll tell you about. So here, and this is all I'm going to tell you about with ACF and Argonaut. If RNA editing is involved, then ApoBec1 and ACF should have an effect on susceptibility. And if microRNAs are involved, then Ago2 should also have an effect. So we tested those. I'm going to show you ApoBec1. These you'll have to be satisfied that the paper is accepted. You get to read about it. It affects cancer risk in a parent of origin manner as well as a conventional manner. It affects cryptorchism and testicular atrophy and infertility, and all of those are risk factors for testicular cancer. So now we have a sense of some of the molecular biology, the genetic basis for some of these effects. But let's look at ApoBec1. We made a knockout on a pure 129 background. Heterozygous through the paternal lineage, across a wild-type mom, regular 129 mouse, father to son to grandson, and we get these 2 kinds of segregants, the wild-type and the heterozygote, Mendelian. 5-fold increased risk. So partial deficiency for ApoBec1, the cytidine deaminase activity, increases cancer risk 5-fold, directly implicating RNA editing in cancer risk, testicular cancer risk. Look at the maternal side, mom to daughter to granddaughter. The same crosses, just done in the reverse way. We get the same genetically identical segregants. It's only the ancestral genetics that's important. None of the sons were affected. So maternal heterozygosity protects the sons. If we cross these wild-type animals together, just like a pure 129 line, that protection persists for generations. It's not 0 anymore, but it's still significantly less than expected. So it's conventional through the paternal line and epigenetic transgenerational effect through the maternal line. So it makes a difference who is heterozygous for a partial deficiency. Now these are just deletions, heterozygosity for deletion. It's not like there's another allele that might be doing something different. This is just a dosage effect that triggers epigenetic inheritance. Then Vicky wanted to know, you can induce the effect, you can propagate the effect, can she reverse it? So she took these males through the maternal line that are 0, and normally you would just keep going. But here she reversed it. Instead of using the daughters to propagate, she took the sons and crossed 2 wild-type animals, the reverse cross. And our thinking, the maternal heterozygosity is what's critical in this case. And if that's the case, this should reverse. but it doesn't. So Vicky was really persistent. She went one more generation of this kind of cross, and then it switched back to wild type, or to the wild type number. And the argument is the maternal genotype has an effect, but it can't affect the fetus because that's already determined. It can't act on the germ cells in the fetus. That's already done. But on the fetus itself, but can act on the germ cells of the fetus that make the F2 generation. She did the reciprocal cross and got the reciprocal result. 14, 13, 0. 2 generations to switch. So paternal is conventional modifier, maternal is a transgenerational modifier, and then we can reverse the effect. And being able to reverse is pretty cool because when we have clues to the molecular biology, we should be able to switch it and show by inducing it and then switching it back that we really do have evidence for the effect. The last part, Kit ligand. This goes way back to Roy Stevens. Roy was not, anyone who's willing to do crosses with 11,000 look at 11,000 animals, you know they're willing to do a lot. So he took all these different mutants at this Kit ligand locus called steal, or the Kit receptor locus called W, and he moved them over some years onto the 129 background, and then surveyed the wild type and W or steal heterozygous kids for tumors. There's the wild type and there's the heterozygotes. Those are the numbers of animals that he surveyed. And it was known at the time that these mutations affected melanogenesis, hematopoiesis, and gametogenesis. Could lead to reduced fertility or sterility. And so thinking about how this worked, maybe these mutations that act during development on the germline could influence susceptibility. But he never published this until Jason did this particular study that was published a few years ago. And the reason he didn't publish it is that here's an example, 7-fold higher in the steal heterozygote versus wild type. Steal J, maybe 3 or 4-fold higher. Steal Dickey, same as background. So why does some steal mutants do have an effect and some don't? And then look at the W mutants. There's no effect. So the ligand has an effect, but its sole receptor in the genome doesn't have an effect, and some ligand mutants do and some don't, and he couldn't make a story out of it. So Jason went through the literature and did some molecular cloning to characterize these different mutations, and it turns out it's the loss of the transmembrane, not the soluble isoform, that makes the difference. And that depending on the particular mutant, you either have that loss or you don't. Then Delphine Carouge, who was a postdoc who just finished, wanted to follow this up. Jason had some preliminary evidence that there was a transgenerational effect. Delphine wanted to follow that up some more. So here's wild type from Roy Stevens in the literature. We always have contemporaneous control. So here's 129, 6.7%. That's not so far off from 7. If we do heterozygous mom to wild type dad, we get steal heterozygous kids and wild type kids, Mendelian, 6.3, 7.1. That's fine. If we do the reciprocal cross, here the only difference is the parent of origin, 3.4 and 14. So this is what Roy focused on. He's got a mutant, a variant, that increases cancer risk. So it's promoting cancer. So you pay attention to that. This one escaped notice until Jason and Delphine worked on it. The wild type animals are reduced. And when Delphine bred these wild type animals together, and we call them wild type because the difference from a true 129 is the ancestral heterozygosity for the steal GB deletion. Reduced. And this is a graphic representation of the fold change. We required both a statistically significant result and at least a 2-fold change, and that's what we saw over these generations. Then she reversed the cross, and it immediately went back to wild type. So we can induce the effect through the paternal lineage, and we can reverse the effect. And it's simple heterozygosity for a steal mutation. Now, paramutation, there's a famous paper a few years ago on this, that particular KIT receptor mutants will induce a change in the wild type allele, and that when you propagate this paramutated wild type allele, you will see white toes and white tail tip, just like you do in a KIT receptor mutant. And that breeds true. And that's the Razul-Zadegan paper. And this was recently repeated in this paper, but there's a really interesting, not a critique, but sort of a critique. If it was my paper, I wouldn't want it discussed that way. But it was done nicely. It's just, But in this case, it's not a paramutation. It's sort of the opposite of that. Because in a paramutation, if you lose the function in wild type, if you shut off that allele, then you reproduce the phenotype that you see in a deletion heterozygote, partial loss of function. Here, we've got a deletion. We get the wild type. So this one's got reduced expression. This is normal expression, the wild type. And here, this is the other allele that was opposite the deletion. But look here. If it's homozygous wild type, 7%. If we lose function, we go up. So loss of function increases risk. How do you reduce the risk? Presumably you do that by increasing the function. So somehow the model is that the heterozygosity for this deletion increases expression from the wild type allele. And in fact, they argue this for some of the other DDX mutations that we talked about earlier, that heterozygosity for the mutation increases expression of the wild type. Now we don't know how that happens. What's the molecular marks? What's the molecular basis? What's the mechanism? We don't know. But that's the model that we plan to pursue. I'm going to skip this. Yep. All I'll say is that dead end is interesting. It acts, if you think about epigenetic inheritance, we always focus on what happened in the parental generation that triggers a change that's inherited, as though the next generation is a blank slate. Nothing is there, and you inherit that epigenetic change, and that's it. We have evidence that dead end, Argonaut2, and ACF act in the offspring generation to increase risk, and they do that presumably by processing the epigenetic information inherited from the previous generation, that there's some biology at each generation that's involved. And dead end, Argonaut, and ACF are involved in that. So to generalize a bit, think about epigenetic inheritance. What do we want to know? You can read lots of cool papers. They focus on particular topics. But if you really want to know the beginning of the story to the end of the story, there's a chain of evidence that we need to have. And this is partly a reminder of the things that we still need to do if we want to understand this new mode of inheritance. We need to understand how an organism, and we know this pretty well, senses the environment and the physiologic state. So we smell, we taste, we see, we touch, we have pain sensors. So all of that's fine. We need to transmit those signals from somatic cells to germline. If it's only in the somatic cells, only in our brain, only in our nose, that's fine. We can smell it, we can taste it, we can see it. But it has to get to the germline in order to be transmitted to the next generations. It's in the germ cell then, it has to be converted into an epigenetic change, because presumably that signaling molecule isn't inherited, but something induced by, modified by, that is inherited through the germline. It has to go through the germline, then at the next generation it has to be converted into a new phenotype. And then you have to decide, work out the mechanisms for persistence or reversion. Now this step is really interesting. When we do controlled experiments, like here where we have a heterozygosity for a mutation, or if we expose a mouse to a smell or to heat or to stress, we have a controlled study with defined animals and we can show the inherited change. But in your lifetime, my lifetime, we're exposed to a lot of stresses. All kinds of stress, things we eat, things we smell. We get a lifetime of experience. How does an organism decide which of those to encode in the epigenetic information, the epigenetic code? And that's laid down as methylation, histones, RNAs, whatever. But then at the next generation, that lifetime of experience has to be translated back into specific phenotypes. It's not just we become noisy. In the cases that are known, that phenotype is recapitulated. And how that happens is completely unclear. We know how DNA does that. We don't know how the epigenetic code does that. But that step from soma to germline, we have a hunch about what's going on. This is the other big problem we're working on. There's probably several ways to do this. This is our favorite. Here's a somatic cell. It expresses the KIT ligand. Here's a germ cell. It's got the KIT receptor. It signals the receptor in the germ cell, and then these are the downstream pathways that we all know and love. We know these pathways really well. So this implies that somewhere through this, there's an epigenetic implication. So I've added onto this the genetic variants that we know that show conventional effects on testicular cancer risk. They act in a conventional Mendelian manner. Those are the ones in red, like KIT here, or this telomerase gene. defect. Somatic mutations, changes that are found only in the tumor, they're relevant to the biology still. So things like these, RAS, GRB2, PNLTC1. And then these that are in red and gray have both conventional and epigenetic effects. And what you see here is that there's a whole lot of genes over here, DND, nanos, PNLDC1, Argonaut, DNMRT. These two pathways seem to be talking to each other. The connections are still a bit tenuous. There aren't so many between them. But we need to work out, are these pathways really talking to each other? Are these integrated in some way? Is this the source of epigenetic information that leads from signaling through the KIT ligand, KIT receptor, to next generations to change cancer risk and perhaps other aspects of biology? So the big questions then, what does epigenetic inheritance and non-random union of gametes tell us about inheritance? We've taken that for granted for a long time. Or heritability. What's the causality and chain of evidence so that we can tell a story that goes from the beginning to end where we know all the molecules and mechanisms in between? How do we recreate phenotypes, as we were just discussing, from changes in the epigenetic code? And then evolutionary adaptations. People in the field, I've written that these epigenetic adaptations solve an evolutionary problem, but they do it in a temporary way, waiting for a good old-fashioned DNA sequence change to fix it, make it permanent, so it's inherited in the conventional way. Makes good sense, except thinking about it some more, I don't understand how it works. Because if that epigenetic change fixed the problem, solved the selective issue, what's the selective advantage for making a DNA sequence change? If you've largely solved it, your work is done. Just make that epigenetic change permanent or figure out how to redo it and you're done. So we need to understand the ways in which epigenetic and conventional genetic change happens in the process of solving environmental and physiologic challenges and leading to evolutionary adaptations. So these are the people that are involved. This is Vicky, did the ApoBec work. There's Jason, who's here, did a lot of the steel work and EIF2S2 work. And that's Gunwa, who did the neural tube work. And Jen, who did the dead end work. And the other people are here, and the funding agencies are here. And this is the last one, looking for postdocs. There's a couple of projects here that could be interesting. So thank you again.
Okay, we have time for 5 minutes for questions. So,
That was really nice, Joe, although every time you give a talk, my head hurts, but in a good way. Do you think, I have two questions. One is for like the last point you made about why would the changes be epigenetic and kept. Do you think it would be a way to be able to like respond, but respond back, like changes that you might need to?
You could be, absolutely, but the trick is, okay, the question, it's me. The trick is, when you make that epigenetic change and you have to decide, do you do this with a DNA sequence change or an epigenetic change, do you know what's coming next? Do you know whether that environment is going to be fluctuated, go back, so you need to revert? You don't know that at the time. It turns out that if the modeling suggests that if things persist, that the strength of selection will eventually make it to a DNA sequence change. But in a fluctuating environment, maybe you spend time doing this back and forth, back and forth, back and forth. But even with that, there's some issues. How does an organism in real time, me right now, you right now, know what's going to be tomorrow or next, and remember it in an epigenetic way? How do we remember we're in a constant versus a fluctuating environment? It's easy to say that. It's nice to model. It's fun to model. But then you think, how would that work? I don't know.
I had one more question. Do you think you were talking about how does the organism sort of uncode the change to cause the phenotype? Do you think you could do some kind of a screen, like where you had a cross like you had and look for mutations where you don't get the response that you expect in the next generation?
Yeah, but you probably wouldn't want to do that in mice. It's hard. The other trick to this, though, and I forgot to mention, all of these genes, these changes, these gene dosage effects induce an inherited cancer risk. We don't know what happens normally. Is there another version of epigenetic inheritance? Are these genes, these effects, part of usual biology? Or are these really mutant effects that are inducing epigenetic inheritance? The normal function is to say, don't do this. We don't know which of those versions is correct.
There are mammals whose eggs don't pause before the end of full meiosis. Certainly other
say that
animals.
again?
There are mammals who don't have their eggs pause it.
Yes.
Have you looked at them?
No.
Can you, or there are no models that...
You're raising a really good point, because now we're at this issue of how do these effects relate to meiosis and ovulation and gametogenesis? And just like this Ottolini paper showed, maybe it's not as simple as we imagine, and you could imagine other species doing something else. We don't know the extent to which these kinds of epigenetic changes are inherited in other species. So it's a really good question, but an open issue.
Especially relative to the womb.
Hi. You mentioned folate had an effect in your model.
I mentioned what?
Folate had an effect in your model. I was just curious, considering commensal bacteria such as Bifidobacterium can produce folate and other commensals as well.
Yeah.
Have you looked at that?
Yeah, we didn't. In this case, we didn't. What you're asking is, can the gut bacteria compensate for folate in some ways? And they absolutely can, and in other studies we've dealt with that. In this case, we didn't design that neural tube study to get at this issue. We studied it and we did it the way you would do it for that purpose. And then we discovered this other issue along the way. Now we need to go back and redo and control exactly issues like that. So, good point.
Joe, with regard to your litter size data, do you know what is rate limiting? If you have a very early lethal, way before implantation, would you expect a normal litter size?
The litter size is largely determined by the number of eggs that are ovulated. And if that egg dies, that litter is reduced proportionally. Now, could that change under these conditions? Sure. But remember, when a female ovulates, she doesn't know the male is coming, the sperm that are coming. So she can't say, ooh, goofy sperm on the way, I'm going to need some more eggs to be better here. That's what the reproductive biologists tell me.
So let's thank Joe for a really thought-provoking talk.